Muy buenas tardes a todos. En nombre del Patronato, Director General y Consejos de la Fundación Ramón Areces, les doy la bienvenida al 6º Encuentro Internacional de la ELA en España. Quería trasladar nuestro agradecimiento a todos los ponentes y moderadores de las sesiones de esta tarde y felicitar al equipo de la Fundación Luzón.
The Luzón Foundation team for the excellent work that they have done in the organization of this meeting. I would like to welcome María José Arregui, who is the chair of the Luzón Foundation, for making the cooperation between both institutions so easy. Cooperation and the promotion of scientific research is one of the priorities of the Ramón Areces Foundation, which has always supported research into rare diseases and neurodegenerative diseases. We believe that promising research is in the crossroads of several fields of knowledge, and that's why we think we need to promote interdisciplinary research. At the same time, we need to implement greater cooperation between institutions and foundations to join forces in different projects. And that's why the Ramón Areces Foundation has decided to support research on ELA, and that's why we're cooperating so much with the Luzón Foundation. And for us, this international seminar, which is now in its 6th edition, has now become a tradition. Of course, we will continue to support the Luzón Foundation and its cause, which is, of course, to find a cure for ALS. Thank you very much. And now I'm going to give the floor to María José Arregui, who's the chair of the Luzón Foundation.
Muy buenas tardes a todos.
Good afternoon, everyone. Once again, we are in the Ramón Areces Hall, and the first thing I'd like to do is to thank the institution, its Director General, Manuel, and everyone in the foundation who've always supported us and welcomed us. And to cooperate with an institution like the Ramón Areces Foundation means a great deal, because it will mean the kind of continuity and depth which we like to see in our efforts. And today we're holding the 6th edition of the seminar in ELA research, which is today to share advances in scientific research and to disseminate that knowledge amongst every member of the scientific community. Because to share information is absolutely crucial so that we can bring together all the findings in the field of ALS. Although actually what we know is still very little. We don't know its etiology or what triggers the death of motor neurons. We only have a few glimmers of light in all this darkness. And we do know some of the mechanisms that are involved, as our speakers will explain today. And that means that every piece of this terrible puzzle is important so that we can have a complete picture which will show us how to put a stop to this, to the progression of this disease. And so in this seminar, we're also trying to make sure that all the hard work and complexity in ALS will reach decision makers so that they can make informed, rigorous decisions, as well as the pharmaceutical sector so that they can cooperate with other sectors, and foundations so that we can learn how best to support scientific research, but especially the patients, their families and associations and everyone who is living with a disease. A lot of work is being done, a lot of very good work is being done with great researchers, great clinicians, great ideas, and a lot of effort. And I think we're seeing a golden age in the field of ALS research. And I want to mention Juan Carlos Unzue and his work with the Barça-Manchester City match. I also want to mention Miguel Ángel Roldán, who has just climbed the Himalayas with the ALS flag. And I also, of course, want to remember all of those activities and actions which are helping us all move in the same direction, all pushing together, focused so that we can make progress. We know that the ALS community is a great team, and we're seeing that in the various regulatory processes that are underway and which we have promoted. This morning at 10 a.m. I had a call from the regional minister of the Galician government, and he told me that they were going to hold a press conference at noon in which they would announce 3,200,000 euros of their budget being allocated for the care of ALS patients in Galicia. And that is no coincidence. It's the result of everybody's efforts, everybody who is a member of the ALS community, scientists, hospitals, associations, foundations, patient groups. These are the results that we are beginning to see. And I do want to mention our ALS units in the different hospitals in Spain, with neurologists, case managers, nurses, and all the health professionals who look after our patients and their families when faced with this terrible disease. And in the foundation, we have a very clear definition of our priorities, and support for these units is one of them. And clinical trials are, of course, the practical demonstration that science is making progress in ALS. And we're going to fight to make sure that there will be more Spanish hospitals taking part in clinical trials. Because when there's the opportunity to take part in a clinical trial, you open a door of hope for the individual patients and for our whole society. So let's support each other with scientific rigor, as you all, of course, demonstrate. But we also need government support, institutions, pharmaceutical companies, associations, health professionals, and patients and their families, because together we are building a future so that ALS will not rob us of our loved ones. Thank you very much for being here.
Buenas tardes a todos. Agradecer a la Fundación.
Good afternoon, everyone. I want to thank the Luzón Foundation and the Ramón Areces Foundation for giving us the opportunity to be in this seminar and to chair this wonderful session, because our speakers are excellent, as we will see. I've been asked to introduce myself, and so I'm going to do that. I'm Charo Osta. I coordinate a group at the University of Zaragoza and at the Institute of Health Research of Aragon for 22 years, as we were discussing over coffee, studying ALS from 2 points of view. We seek new strategic strategies. I have a degree in veterinary science and a PhD in veterinary science in animal models. And of course, the team is also searching for biomarkers that can be used for diagnosis and to define the prognosis based on different products in cooperation with many hospitals, specifically a very close partnership with the 12 de octubre hospital in Madrid, which from the animal model is extending it to patients in cooperation, particularly with Dr. Garcia Redondo. And of course, we would all love to have an effective treatment as soon as possible. I think there is some way to go, but as María José was saying, we're seeing greater efforts made, and that's a good sign. And in order to achieve the effective treatments we're all seeking, you sometimes also need to understand the mechanisms of the disease and understand it from different perspectives, from basic science, which, although initially might not seem to provide short-term results, but in the future, there could be therapeutic targets that might be identified and which could be useful. And I think that's what we're going to talk about today. We're also going to hear about applications, but mostly about some basic research, which is increasing our knowledge of ALS. And I'm not going to take up any more of your time because you're here to listen to the speakers. And our first speaker is Professor Robert Baloh, who's got a degree in medicine and a PhD from the University of Washington, who completed his residency in neurology at Harvard in Boston. He then went back to the university as a lecturer, then worked there until in 2011 he traveled to the Cedars-Sinai Center in Los Angeles, where he's been doing extraordinary work in basic research in ALS, frontotemporal dementia, muscular dystrophies, and other entities. He led that team until he changed course and went to the private sector. He worked for Roche and is currently global head of neuroscience of the Novartis Institutes for Biomedical Research. Also Bob, if I may call you that, or Dr. Baloh, is so amazing that he's actually going to give his lecture in Spanish. That's definitely something to thank him for making the effort. Go ahead, Robert.
Okay. Buenas tardes a todos. Sí, es verdad.
Good afternoon, everyone. I've given this talk 1,000 times in English, but it's the first time I'm giving it in Spanish. And so I hope that you will be lenient. But it's a real pleasure to be here and to speak about my research in the last 15 years in the neuroinflammatory mechanisms of neuronal injury in ALS. Topic very close to my heart, and this is me in 2005. I'm a doctor, as you've heard, and this child had a different type of motor neuron disorder, and he was in a children's camp. And this is me also in 2020, before I joined Novartis, which is where I work now. And the story here is that after maybe 3 years, since about 2016 to 2020, 3 treatments were found for some which could cure the disease. And that had a great impact on me. And so I decided after that, this was my very last patient in Los Angeles with ALS, I decided to move to Switzerland and spend all my time trying to discover treatments for ALS and other neurological diseases. Well, this is the summary of my talk. There's going to be 2 parts. First, I'm going to talk about the genetics of ALS and how it involves the immune system and the inflammation, and how it affects the injury of motor neurons. And then the second part, which is really new, we've just published this clinical trial in medicine just a month ago, and it's a clinical trial on stem cells in ALS. It took us 10 years to do this. And the attempt to reduce inflammation and prevent the loss of motor neurons. First, what is ALS? Of course, I don't need to tell you, but as you know, there's a loss of upper motor neurons in the brain, but also lower motor neurons in the spinal cord. And there's a lot of symptoms, which include muscle weakness, muscle atrophy as well. And there's this, which are fasciculations, just something that's hard to pronounce in both English and Spanish. And that's when motor neurons are irritated and they send a lot of signals to the muscles. And so that's ALS. But I have to mention a case because the disease doesn't just affect motor neurons. This is one of my patients who was a former NFL player, so it's American football, and then became a successful lawyer. And he started to develop first a cognitive deficit and changes of behavior at the age of about 50. But then after about 10 years, he started to develop some tremors in the right hand and muscle atrophy, and then after that, muscle weakness that progressed, accompanied by those fasciculations or muscle contractions all over the body, and the weakness extended and involved the other hand, and then difficulty swallowing, breathing, legs, and eventually in 2015 he was admitted to a hospice and died soon after due to a respiratory failure. This was his brain, and you can see for two things. First, there's atrophy. I don't know whether you can see it, actually, in the frontal lobes, but also in the temporal lobes, which means that he had another disease as well, which is called frontotemporal dementia, which was mentioned earlier too. And so there's a spectrum, actually, these two disorders which overlap. And ALS, as I said earlier, affects motor neurons, whilst frontotemporal dementia affects certain parts of the brain. But there's considerable overlap. And so up to 50% of patients with ALS also have some destruction of their frontal lobes. About 15% have actual frontotemporal dementia. But 50% of patients who have frontotemporal dementia as a diagnosis also possibly have ALS or have a confirmed ALS diagnosis. So it's quite a big overlap. And here on the right-hand side, you can see the genes which we discovered in the last 15 years or so may be involved in both. And in most cases, it's both, not just one or the other. And so I'm going to focus during this part of the talk on the genes here in the central area, particularly C9 or 72, which is really hard to remember in either language. And I'll explain how this gene works and how a lot of these genes work. They act on this part of the cell. They're involved in endosomal, lysosomal traffic. This is the system to ingest things from outside cells, but also to break down things inside cells. And this here, this gene, or the protein that's coded by that gene, the C9orf72, but also all the genes in red may cause ALS or frontotemporal dementia also. And we know that this pathway is extremely important, but we don't know exactly in which cells and why, and what happens that might eventually trigger the disease. And so I need to say more about this mutation, which is very specific in C9orf72, because that's a repeated expansion of this region here, 4G, 2Cs. In normal subjects, there's only 2 repeats, but in patients, you can have up to 1,000 of these, which is a lot. And there's 2 effects of this mutation, to the right side and to the left. It can cause a reduced expression of the gene, and in patients who have ALS with this mutation, there's a reduced activity of about 50% of the protein and also the RNA in the... But it can also cause other things if the gene transcription starts in front of that mutation. It can be incorporated into the RNA, and that may add or augment the activity and pass to the nucleus of the cell, but it can also become translated into proteins, and those proteins may also accumulate in the cells and cause damage. And so there's a lot of these processes, and there have been a lot of publications describing a lot of these processes, but it's interesting because 6 months ago, there was a clinical trial using antisense oligonucleotides made by a company called Biogen, and in that trial, they were able to reduce the accumulation of these toxic gene products, but there was no effect or no observed effect in the patients. So there was no slowing down of the progression of the disease. So before and now, we are focusing on this, on the effects of this deficit in this gene. And during the rest of the talk, I'm going to describe this, which is the research that my lab is doing in this area. And so initially, we obtained some mice without this gene in order to see what would happen. And we expected the mice to develop problems in their nervous system, but that actually never happened. What they did was to develop immune problems, and that surprised us. And here in this big boson, which is 3 times the normal size, when we looked into what had happened, we found some cells inside the boson which looked like macrophages. Macrophages had various scavenged materials inside them. And when we looked at what had happened in the cells, we saw that the lysosomes were engrossed, and so there's a problem with this system. And also there were some abnormal immune responses, exaggerated immune responses, which made this seem like an autoimmune disease. But why? And here you can see that the dendritic cells, some macrophages and also the glia cells in the brain, but there are these cells that express, I don't know if you can see it, C9orf72 more significantly with dendritic cells. And so we decided to look into what was happening in the dendritic cells. And these are actually extremely important cells for the immune system because they're a sort of thermostat for the immune system, for T and B lymphocytes in order to fight mutations and cancer too. But if that thermostat is too high, is set too high, you can induce an autoimmune response because it will not allow for tolerance of endogenous antigens. And so in our lab, we looked into this with this researcher in the photograph from our team, and she found that in the dendritic cells of very young mice, before they developed that autoimmune disease, in the dendritic cells there were problems with the RNA sequence in this system to defend the cells from virus, particularly if interferon type 1. There was exaggerated response, and you can see this here in every one of the mice that were knocked out for C9orf72. We can see all the genes in this response are overexpressed. And she then looked into the why of that, and it seemed that initially, she stimulated the cells with agonists for the pathways involved in fighting bacteria or virus, and most of them were normal, but the response of the agonist for this pathway and this protein, which is called STING, was really exaggerated. And so you can see here that in the mice that have the mutation, this very elevated response in comparison with normal mice. And so what is this pathway? This is a pathway which detects DNA nucleotides, typically a virus, but also mitochondria and other cell damage. And this is a pathway which, after a couple of other protein steps, induces the expression of genes in the nuclei which fight these infections or destroy these fragments. And that causes a lot of inflammation. And we knew already that lysosomal system is very important to put a stop to this signal, to degrade this signal. And this is what you see here on the right. After it's activated, it needs to be transferred to the lysosomes. And if not, that's going to produce an exaggerated response of this, or activation of this pathway. And that's really important because, as I said before, this endosomal pathway is affected when C9orf72 has a mutation. So that's why we focused on that initially. And of course, there was a delay in the breakdown of STING, and so it persisted in the cells after it was activated. You can see this up here. After 24 hours, generally in normal mice, without the mutation, there's no longer any STING expression. In the mutated mice, however, STING is still there. And so our theory, our hypothesis, is that normally STING travels from the endoplasmic reticulum and Golgi body, and then it has to break down through autophagy and through lysosomes. But when this gene is mutated, that doesn't happen, and so there's an accumulation and a sustained activation. That was the theory, but we decided to design an experiment to prove that. And we obtained STING by crossing mice that were CD72 knockouts with the ones that were STING knockouts. And what happened was that there was an increase in the size of the boson and also of the response of splenic cells to interferon 1. So almost all of this response, this elevated immune response, is mediated by this. Mutation. And of course, it might seem as if mice would have been more susceptible to autoimmune diseases because of this. And so again, it was that thermostat I mentioned on the right-hand side. But why, and what does this have to do with tea in China, as they say in English? And this is because it's true that patients who have ALS also tend to have a higher prevalence of autoimmune diseases.
We knew this before, but we didn't know why. So in ALS here, and especially in patients that have C9 mutations, that have frontotemporal dementia, and also in ALS. And sometimes patients that can have both diseases, multiple sclerosis and ALS, it's very rare, but in this group of patients, 80% of them have this C9 or RF72 mutation. So it's very interesting. So we decided to do an experiment in mice too, to see whether they are more susceptible to a model of multiple exposure to experimental autoimmune encephalitis. And here you see in the mice that are missing 2 copies of the gene have a much more severe disease than the mice that are only missing 1 copy. and the normal mice. So humans, like humans, possibly there's a propensity to having autoimmune diseases, but they need a stimulus, something in the environment to cause them. So mice are not humans, so what is this situation? Well, we look in patients, in the blood of patients, we searched and looked and we isolated macrophages in the blood of patients with C9 ALS, 5 patients who had ALS from other causes and 5 normal patients, and we saw exactly the same thing. The dendritic outlets of the mice missing C9, there's an activation of or response, type 1 interferon response, and only in the patients that had that mutation. In the other patients, this did not happen. There was not this response. We decided to repeat it in more patients because we'd only used 5, and a friend of mine in Colombia had about 259 patients with ALS and 20 with C9 ALS, and you see here, because we did the RNA sequencing, that there is a decrease of the scattering of the gene of about 50% in the mutated patients, but exactly the same response, exaggerated response of interferon type 1. That happens in patients. And finally, here, we tried to rescue this effect, to do something about it, and we did it in 2 experiments with PBMCs, the patients with the C9 mutation, and this is not a human medicine, but it can be used to investigate MX1. And we saw in 2 experiments blood macrophages and also another experiment, macrophages that have been cultured for some time, but in each different situation we saw the same. So it's possible that we could use, we could reduce this response in these patients in this way. And this is very important because it goes back to what I said before about the peripheral immune system. But also in the brain, there is evidence that this route, that this response is very marked. This is the gene expression, but these are the routes, the pathways in the cerebellum in this case, but also in the brain, I believe, that are overstimulated. And this is viral response. This is type 1 interferon response. I've almost finished this part. How long have I got left? I don't know. Let me go into the next section. What are the myeloid cells in the brain that are causing this situation? Because we investigated and researched to know what happens in the brain, because it's very interesting. Brain cells that express C9orf72 are the microglia, not the neurons. So we researched this, and this is very interesting because the microglia, were discovered here in Madrid, very near to where we are. And Cajal, they were discovered by Ramón y Cajal, who discovered there was a third element, there was, and it wasn't the neurons. So he tinctured these samples, and he could see the neurons, but he couldn't identify the other bodies. And then Rio Ortega invented a new way of tinting these samples, and he discovered that there were other types of elements, the microglia and the oligodendrocytes. And this was in 1919, when it was seen that they were like macrophages in the brain, and they could respond to damage and infection exactly in the same way as in the peripheral immune system. So we researched the effect of C9ORF72 in microglia, and it was almost exactly identical. The response to sting stimulation was very, very marked. And this is the degradation of sting, and it's delayed also in the microglia. And this is the RNA sequence in the immune system, and we see how in the peripheral immune system the interferon response genes, type 1 interferon response genes, were really increased very much. So what happens here? What do the microglia do? Well, they fight damage and they fight infections, but they are also responsible for the degrading of the synapses during development and also during aging. So we found that in mice that are missing this gene, there were less synapses, more complement, which is a part of the system, the degradation system, and this is produced by the microglia. And also the mice were not normal, so they did not develop ALS, but they had brain problems with their memory, things like that. And we believe that they have this propensity, but we don't know exactly why they don't develop ALS or dementia. They have problems with their memory, mainly. And there are 2 slides for this part, and then I've got the final section, and then we're done. Now, this is the story. The microglia have problems in the endolysosomal function and a hyperactivated response because of the type 1 interferon response that is driven by sting, we believe. And this causes a loss of the homeostatic program, the deposit or accumulation of complement, and it causes a reduction in the density of synapses as we get older, which is something, that word in Spanish I find very difficult to pronounce. Aging, envejecimiento. And now, I've spoken about this gene C9ORF72, but there are other genes involved, and this is the final slide of this section of my talk. We believe that many of the mutations that can cause ALS arrive through their effects in the disease, happen through the myeloid cells because they work perfectly well and they're designed to respond to the environment, infections, attacks, football, and cause problems in homeostasis of the brain. So the effects are clear in the peripheral immune system. They're not clear, sorry, but we're investigating that. But we know it has something to do with neurodegeneration, and we can use drugs or other things to reduce the effects in the myeloid cells and the microglia and see whether we can delay the development of the phenomenon of the disease. And this is the summary of the first part. I've only got 5 slides more. We did a clinical trial to try to use stem cells to reduce inflammation in patients who have ALS and rescue the loss of motor neurons. And this is what I said before. We've just published this in September. And the theory was that we could replace a type of cell using stem cells and turn them into astrocytes and supply GDNF for the motor neurons in the spinal cord. So this is the intact nervous system, but with ALS there's a problem with the motor neurons, and also with the astrocytes, and also with the microglia that are not in this slide. But my colleague, because this was not an idea of mine, my colleague thought, well, if we can introduce new astrocytes, maybe they can save the motor neurons. And there are many effects that can be used to achieve this. He created a type of stem cell that was genetically modified to release this trophic factor, GDNF. And in mice, because this on the right is the spinal cord of a rat. It's an ALS model. On the left, so he and I, injected the stem cells. We call them CNS10 in this case, MPC, GDNF, on one side of the spinal cord. Where we injected them, and these are the cells, and these are the motor neurons. And the side that had the implant, the product in it, in that area, the disease did not develop. But where we didn't inject the cells, the disease did develop. So we tried to do this in a clinical trial in humans. This was 10 years ago. And in humans, the cells become astrocytes, and they are designed to express this GDNF. And this is what we did with 18 patients. It was a phase 1 and 2 clinical trial. That means that this was because of safety concerns mainly, but also to see if it was dose dependent. And these injections were similar to the injections that we'd given rats. We'd done it unilaterally. We spoke to the FDA, the authorities, the FDA in the US, so that they asked for authorization, and they said that there couldn't be a placebo group used because we would have had to do false surgeries and whatnot. So in each patient is their own placebo, but on the other side of the spinal cord. So we compared them to themselves, and we did multiple measurements of leg function to see whether there was an effect on the spine of the stem cells in the survival of the motor neurons on one side of the spinal cord. And the neurologist, I was the neurologist, and the patient, we looked at the side where we implanted the GDNF to see, and we were the only ones who knew, the patient and myself. This was the first patient treated in May 2017, and we had to invent a new device to inject the cells in the spine. It's a very, very small space, just mere millimeters, and it was very difficult. And then after this, it looked as if this was safe to do. Inject 10 times in the lumbar spine, and there were no problems with the patients, tumor, inflammation, no problems. But after the injection, there were no problems. And this is a picture of a volunteer sitting in this chair to measure the strength of her legs and arms. And before we did this, we did an investigation to see whether the progression of weakness on both sides of the muscles in patients with ALS is similar. And it is. There is a high level of correlation. The strength in the arms, in each muscle of the arms and the legs, there's a high correlation in that level of strength. So that's why we thought we would look at both sides and see if there was a difference between one side and the other, and if there was, whether the stem cells had made any difference. But unfortunately, it was safe, this procedure, but there was we didn't see any effect. There was no effect. So the 18 patients, the rate of demineralization was the same on the treated side and the untreated side. And there's another way of looking at the data. There was an effect, a small effect, and this is the treated side compared to the untreated side, and both doses. If the data that at the top, above the line in the middle, there's a rate of demineralization which is slightly lower than on the other side. So there was a slight effect, but it was not really significant. And this possibly was the reason. And with this clinical trial, it took us about a year, but we followed the patients until they died from ALS. And we recovered 11 of the patients to do the post-mortems to see what had happened with the cells and the motor neurons, and to see what we're going to do or we could do in the future. And in this case, we see that in each of these patients, up until 42 months later, the cells survived for up to 42 months, which is very interesting. It's longer than in any similar previous clinical trial. And they're tinted for GDNF. And you see that in many patients, the position of the graft is slightly, is very much displaced to the top and to the side and far from the motor neurons. And possibly that is the reason why there was only an effect in a few patients, but not in all of them. And we've got to improve our grafting technique. And another thing was that there was an effect of GDNF, which you see here, because it's easier to point out on the slide here. This is a tumor, a benign tumor of the Schwann cells, which probably was affected by the GDNF, by the cell response in the face of this factor, this product, GDNF. This is the end. We also looked at how initially the experiments in rats, because we did them to compare the number of motor neurons on either side and how strength, there was no difference in terms of strength. And these are the conclusions. So we found that we could do this, and it was safe, and in general well tolerated, which is very interesting. But there was a bit of pain on the side that was grafted immediately after surgery, but it didn't last long. So that was the only thing that happened. But demineralization and the reduction of the strength was similar in both legs. And we had the opportunity to analyze the tissue. We saw that the course of the disease was not really altered or improved. And by the way, that was decrease all the time, not demineralization, but disminution, decrease. Good. This slide is about, you see a picture of a visit from Professor Hawking, Stephen Hawking. 10 years ago, he came to visit us, and this is my colleague who designed the stem cells that we used in the trial. And we were talking to him about the experiment and what might happen in the future. And Hawking said, well, his assistant said, he's got something to tell you about all this. And the comment he had was that he said, this is very exciting, but I'm not holding my breath, he said. This is very exciting, but I'm not holding my breath. Good. And this is the end of it. These are the acknowledgements. I have to thank all the people in the lab and Cedars Sinai, and also the other labs, collaborators, co-workers, and the clinical trial team that was very large. And the experiment was fantastic. This team is continuing with the research, the experiment, to try to improve the surgery technically and also to try to put a graft in the brain, which will be the next thing that we will try to do. So thank you very much for putting up with my Spanish.